Hi, I'm Chance.
And I'm Sarah Catherine.
And this is Conservation Connection.
Presented by Last Chance Endeavors.
We run a wildlife education nonprofit focused on connecting students to their environment.
Each week here on Conservation Connection, we do just that by introducing you to the groundbreaking science and conservation work that's happening every day around the country.
We talk to professionals in the world of conservation science and wildlife management and ask them about their career,
their current projects,
their wild and crazy stories from the field,
and everything in between.
Join us each week to discover just how these dedicated people are working to protect our planet. Alrighty guys, welcome to another episode of Conservation Connection. We are back here at Grace Marine Lab working with this year's group of research experience for undergraduate students who are doing some really cool stuff here in Charleston this summer, 2021. We're sitting here in the room with Adriana, Sophie, and Emily.
Welcome to the show, guys.
Yay! It's okay. Sometimes people don't say anything, and we're like, all right.
So we are so excited to be here talking with you guys. We're going to do like the 10-second, what are you studying this summer? Let's start here with Adriana.
So I'm studying the effect of algae on microplastic distribution in stormwater drainage ponds here in Charleston.
Cool. And Sophie?
I'm looking at the delta N15 values and the heavy metal content in seaweed found in Charleston to see if it's safe for human consumption.
And we're going to unpack that quite a bit. And Emily, what are you studying?
I'm looking at how temperature affects horseshoe crab blood.
All right. So I feel like we got to start by unpacking a little bit about the delta N15 value. What is delta N15?
Ooh, starting off with the hard one. So basically, it's looking at stable isotopes. So there's the delta N15 value, which basically looks at storage of nitrogen as well as the source of nitrogen, so where it's coming from. The different values represent different sources and different content of nitrogen. So the lower ends of nitrogen are going to be either your atmospheric or natural amounts of nitrogen, or it's going to be fertilizer. And then the higher amounts are going to represent sewage water.
Okay, cool. So it's using stable isotopes is a little bit like using ink bombs in bank robberies, right? Where it's a way to track where that money has traveled. Except in this case, we're tracking where that N15 is moving around as it gets incorporated into different types of molecules. So you can find it wherever it ends up and figure out where that organism was pulling its nitrogen from, right?
Yeah.
So what made you even want to study this? Have you just always been really interested, or did you have like an epiphany in a class and like, oh man, this sounds great?
So I'm actually coming from a public health major, so most people in this program are marine biology majors, and I have always been fascinated with environmental health and the intersection between environmental health and human health. And I actually am most interested in agricultural sustainability and finding different ways that we can produce food but also have less of a footprint on the planet. So everything I do is pretty much motivated by food and eating, which I love to do. But I read this book by Bren Smith, which is called Eat Like a Fish. Super good, super enlightening. And he coined this term, climate cuisine, which I really liked the sound of that. And to me, that meant adapting our food sources or our food systems with this changing climate to have less of an impact. So, and his way of doing that was through farming seaweed. And seaweed farming is a really great option because it can take up different effluents in the water, so like nitrogen from fertilizers and also phosphorus from fertilizers, and also it can uptake dissolved carbon from fossil fuels, and it uses those for its energy and storage. And then it can also, so it can take up these nutrients and clean and filter out the oceans, which is great. And then also it can be used for all these different purposes. So you've got food, which is what I'm most interested in, but can also be used as animal feed. There's studies that show that it reduces the amount of methane that cows fart.
Which is a huge problem.
Yeah, yes, definitely an issue. So that's one great thing that it can be used for. It's been looked at for biofuels, cosmetics, so it has a lot of different purposes. But I favor food because just everyone, we like to eat. I like to eat.
Yeah.
And it's in a lot of different food products that you wouldn't even think about, like ice cream, beyond just like the sushi, what you like see. So.
Which is also delicious.
Which is amazing, delicious. Love me some sushi.
I love it. We're gonna come.
Yeah,
we're gonna come back to this, but we're gonna spread the love a little bit here. So I want to hear a little bit more about how algae and microplastics in storm drains. What is that whole world? Tell me.
Yeah. So, a lot of people know about plastic pollution in the ocean, especially the big stuff you see, giant garbage patches. But the majority of plastic pollution is not visible to the naked eye. So I'm looking at pieces of plastic that are less than 5 millimeters in size, typically less than a millimeter in size. So in these stormwater drainage ponds, these microplastics get flushed from land into these ponds by big storm events. We have them all the time here in Charleston. And these stormwater ponds let out the water into the creeks and rivers slowly. And while the microplastics are in these ponds, they're going to be interacting with everything else that's in it. So algae is a huge thing here. And these species of algae can emit exopolymer substances, which are basically just substances that they excrete that can affect their environment and the things in it. And a lot of plastics are really susceptible to being changed by these substances. Their binding properties, I would say. And they'll start clumping together. So these pieces of plastic that are so small that you can barely see might clump into a piece that's big enough to sink and hit the bottom of these ponds and then stay there rather than being flushed out as the water slowly drains into these rivers.
So are you creating this in a lab, or are you actually going out and finding these microplastics and these clumps that you're talking about? How are you going about studying this?
Yeah, so I'm doing kind of a two-part experiment. So I'm taking field samples, which basically just involves me getting on a kayak and paddling out to these little ponds and taking samples from the surface and from depth. And then I'm also creating an in situ experiment where I'm creating these little miniature ecosystems in these buckets where I'm going to put them in the ponds, let the water filter in and let the algae get in these buckets. And then I'm going to dose them with a known amount of microplastics that I know whether they're supposed to float or sink, what color they are, all these things, so that I can then, after 2 weeks, take samples from this and actually see exactly how many floated, exactly how many sank, and if the algae is actually having a big effect on their ability to deposit onto the ground.
And are microplastics so prevalent that you can just go out in your kayak, scoop up some water, like stuff off the bottom of the ocean, and you have some?
Yeah, this is going to be a little depressing, but microplastics are literally everywhere. The most common type are fibers, plastic fibers. They come from synthetic clothes. When we wash our clothes in the washing machine, all these fibers shed. They get flushed out into drainage facilities, the places that manage your water, and then a lot of them end up in the ocean. But there's a statistic that says that there are more pieces of microplastic in the ocean than there are stars in our entire galaxy.
Wow.
Which is a little unfortunate.
Definitely.
That's rough, yeah. But it does mean that there's a very big need for people who are looking at ways to reduce microplastics, and that seems like it's you. So
fingers crossed.
Yeah, that's kind of a really... Would you say that storm ponds sort of represent the first step in the transition from land-based to the ultimate residing place in the oceans, for the transition of plastic from the land to the oceans?
Definitely, yeah. Especially places like here, right along the coast, every single one of our storm ponds is going to connect to a tidal creek or river. So it is direct access straight from the land to essentially going straight to the ocean. And the majority of microplastics actually come from land. They're terrestrial based. Tire wear particles are a huge problem. A lot of people don't even realize that their tires create these microplastics, but they are synthetic and they will never go away. And they're becoming a much bigger area of study because obviously all the roads here go over bridges. These bridges deposit directly into the water as well. So yeah, definitely.
I mean, they're so small that we can't see them. So what problems do microplastics create for people?
Yeah. One of the big issues is people love seafood. It's one of the biggest sources of food and also like a economic boom for a lot of places around the world. but you catch a fish and you eat it, and you are ingesting these pieces of plastic that are in them because they're ingesting it as well. They filter water all the time. So these pieces of plastic, they can emit chemicals. There's a lot of toxicity studies around BPA and other phthalates and chemicals that these plastics are made with that can interrupt endocrine systems in humans. So for females, that's hormones, especially along with fertility in women. In animal species, same kind of thing. It affects their ability to have babies. If they have high enough levels of these plastics in their body, it can be so toxic that they'll just die, especially in organisms that are filter feeders. So here in Charleston, everyone loves a good oyster. They filter feed like crazy, and they're one of the biggest areas of study for microplastics because of that.
Yeah, we had some oysters last night, so.
We each only had one oyster.
Only one.
Only one, which I think I don't know. Maybe you tell me, can I have an oyster?
Yeah, I mean, you're not going to die.
You do what you want, Chance.
Fair enough. Okay, cool. We're going to come back to that too, but there's somebody else that I want to hear quite a bit more from.
Yes.
And,
Emily, please tell us about horseshoe crabs, because, I mean, I love the algae, I love the seaweed, microplastics, I don't love them, but I appreciate you studying them. But I am much more like a big animal, big scale kind of thing. So yes, horseshoe crabs. What you got for us?
All right, so I'll just start off with some stuff about horseshoe crabs. So horseshoe crabs, they've evolved little in the past 450 million years. That makes them older than trees. They're more closely related to spiders and scorpions than actual crabs. So if you look at the underside, it looks like just like a scorpion with like a big outer carapace. Kind of creepy looking, but
they're terrifying looking. They look like they would hurt you really badly, but they're so sweet.
I mean, they pinch a little bit. It's just
they're just trying to hold on.
Yeah. But so essentially, they have a really unique circulatory system. Instead of blood, they have hemolymph. That's what it's called. It's blue because they don't have hemoglobin. They have hemocyanin. And they have a unique immune system that is comprised of their primary blood cells. So these blood cells are called amoebocytes. And in the presence of bacteria, they clump up. So it's like a smoke detector. If it detects smoke, it beep, beep, beep. Humans found out that these clump up, then it'll find the bacteria, and we use it to test our vaccines or IV fluids. So there's a huge human industry for horseshoe crab blood.
Because we can't replicate what they already do so well. Like, we can't synthesize the same kind of stuff that they just make as their blood, right? That does the same thing in the same way?
There is some synthetic, but it's very expensive to make and less accurate. I guess some people are probably working on developing it further, but right now, horseshoe crab blood, it can detect like such little quantities of bacteria per amount of horseshoe crab blood. It's really effective, so we use that more often.
So it's basically the way that we make sure that our medical products that are about to be put into a human body don't contain foreign pathogens that are going to make somebody sick, right?
Yes, exactly.
Cool. So that's sort of the baseline on horseshoe crabs, but I don't think you invented horseshoe crabs.
No, I wish I did. They're kind of cool.
They're pretty awesome. So tell me what you're doing with them.
So when these crabs are harvested to have their blood taken for the biomedical industry, they are held in small holding ponds. And these holding ponds can get really hot or really cold. We don't know much about the temperature, but we just know since they're so small, it's not really replicating the ocean ecosystem. So we are looking at how horseshoe crabs respond differently to different temperatures. And in doing this, we will hopefully be able to identify if crabs are held in really hot water, their blood might be of a lesser quality. And if they're held in really cold water, who knows what'll happen. But we essentially want to maximize the quality of crab blood collected while minimizing the actual amount of crabs removed from the ecosystem, because when they're returned to the ecosystem, they have up to a 30% mortality rate.
Right. Yeah. I don't know if anyone has given blood, but like once you take blood for the next couple hours, you're a little woozy. There's a reason they make you sit and like drink juice and eat a cookie, because like you just had very important chemicals drained out of your body. So I can imagine that it would be a million times worse if you were abducted by aliens and held in a hot holding tank first and then released into the ocean with sharks.
Yeah, and they take up to a third of their blood. They only take a little bit of our blood.
Right.
Can't imagine if they took a third of it.
Right. Yeah. So it's both a study on trying to maximize the quality of the product that we're extracting from the horseshoe crabs, and also ways to minimize the environmental impact we have on removing these individuals from their environment and then returning them to it afterwards,
Yes.
right?
Exactly. And this is a huge industry. A gallon of LAL, I think it's the 5th or 6th most expensive liquid in the world. A gallon costs $60,000.
Oh my gosh.
This isn't just straight crab blood. It's not like you take the crab blood and then it's like, oh, it's money-making liquid. But it's once it's processed into limulus amoebocyte lysate.
And that's the medical grade product.
Yes, exactly.
How many crabs do you have to collect from to get a gallon of that?
The REU students actually did this calculation. So we collect 5 milliliters of crab. So I think that's about 80 crabs or something like that. But it comes out to being $89 per 5 milliliters.
Wow. Okay. All right. That's still, that's still pretty good.
Yeah.
So what have you found so far? Have you made any discoveries on what your research question is?
So far, I've only looked at 2 tanks of the crabs acclimated to 30 degrees Celsius. So I don't have anything to compare it to. But we've also been taking their heart rate. And I don't know, I've never seen another paper about horseshoe crab heart rate. So it's kind of cool to take the heart rate and be like, oh, has anyone ever seen this before?
Do you have like little tiny stethoscopes?
No. Like I said earlier, it reminds me of recording a podcast. I have a multi-channel recorder that you plug in the various infrared heart rate sensors, and you fold the crabs in half, and it exposes this small part of their heart. It's like a big vein, and you just put this little sensor on it, and it can pick up the heart rate with an amplifier. So yeah, it's a lot like the music recording business.
That's awesome. So I want to know a little bit about where are you guys from before here and what brought you to Charleston?
Let's start with Sophie.
I wish I don't have the best answer for you. I just found the marine biology program, but in general, what brought me to Charleston, I came from a landlocked area, so being on the coast was enticing. But other than that, I had a feeling. I kind of went with my gut on that one and just came here. That's pretty much the best answer I can give you for that.
I love that.
That's what happened with my undergraduate too.
That's a great answer. What about you, Emily?
So I was also landlocked. I grew up in the Midwest, and I don't know. I was between another school in the Midwest and College of Charleston. And at the end of the day, I was like, College of Charleston's on the coast. The other one is prime polar vortex area, I think.
The fun stuff.
Yeah. So I was a little sick of freezing. I decided I'd try out hurricanes instead.
Yeah. Just a little change of pace,
Yeah.
Still extreme weather, just
keep
totally
me on my
different.
toes.
Yeah, there you go.
What about you, Adriana?
So I'm from Virginia, and I've known I wanted to do marine biology since I was probably 6 or 7 maybe. And so when I was looking for schools, I only looked south of Virginia, didn't even look in state, and I found College of Charleston 2 days before the application deadline. And I said, why not?
Yeah.
Might as well. I already applied to 6 other schools.
Life isn't stressful enough.
No. And I came down to visit 3 schools in South Carolina. This was my last stop, and I got here and I just knew that it was going to be good for me. So I was like, this is it. This is where I'm going, and I do not regret it.
Good. I know this is going to be an anxiety-inducing question, but any thoughts on where you guys want to go next? Obviously, we're still a ways out from that, but where would you want to go next?
Well, Sophie, you're about to graduate. Do you have any thoughts on it?
You'd think I would know a lot more than I currently do. But I do want to go to grad school at some point. I think I'm going to take a break before I do. But I was looking over in California. They have a program, or just environmental management programs is mostly what I'm looking at, or coastal management. So that graduate school at some point, but I would love to travel and potentially do some internships if I can, still keeping agricultural sustainability theme and continuing on with seaweed aquaculture or integrated multi-trophic aquaculture, which is a lot to unpack too, but I can explain that later.
Yeah. Well, actually, I kind of want to go into it now, if that's
Yeah.
okay with you.
Let's do it.
Yeah, sure.
Because, seaweed farming, I feel like, at least, we've said this in another episode already, like, it's a hot topic in marine sciences right
Definitely,
now.
yeah.
For any of our listeners who haven't already heard about it, we're going to get into it some now. Why is it such a big deal?
Seaweed aquaculture itself, because it can filter out some of the effluents in the ocean, like I said before, so like nitrogen, phosphorus, carbon dioxide, dissolved carbon dioxide. So that's great. And then also it has all those uses. But if you pair it with different organisms, so integrated multi-trophic aquaculture is basically combining different levels of the trophic system. So starting off with the autotrophs seaweed, which are like the filter feeders as well. They filter out like the nitrogen and phosphorus out of the water and use that as energy and fuel. And then going up to like the bivalves, so you got your mussels and oysters, which we have here. And those are also acting as filter feeders for similar contents, the inorganic nutrients in the water. And then you move up to like finfish, and so those are like your groupers and other larger fishes. And then if you combine that, you're creating like this reef system, and then also decreasing the amount of waste that's going in, because like your fish poop, the nitrogen in that is going to be used by both the bivalves and the seaweeds. So combining that is a really great sustainable option.
So it's sort of like those self-contained aquaria that you sometimes see where it's like a glass bubble that's got some algae and a shrimp or two, and then it's actually completely sealed up. And those nutrients just cycle over and over because the shrimp eats the algae and excretes waste, which goes out into the water column, which gets uptaken by the algae to grow more algae, and it goes around and around and around. Except it's on a much bigger scale to the point where it's helping people because it's producing a harvest and a livelihood for the aqua farmers, right?
Exactly.
That's like, if I were to retire and just like have my own little place, that's totally what I'd be into.
Come seaweed farm with me.
Yes. Yeah. We would love to do that. That'd be amazing.
Do you think you would like to start a seaweed farm?
I mean, somewhere down the line, yeah. Or like just like the integrated multi-trophic aquaculture system in general. They do it much more up north, so that's kind of why I wanted to study it down here. We have different species, so up there they usually use up in like Connecticut area, that's where it's starting to get big, and just they use kelp, which we don't have down here because our waters are a lot warmer, but we have other species. So I'm looking more at like the local native species, as well as like we have an invasive species that I'm looking at that's already here. So
we haven't asked this question yet, but how edible is the seaweed in Charleston?
That's what I'm trying to figure out.
Okay, so to be determined.
To be determined, exactly. So basically what I'm doing is also kind of like a water quality analysis at the same time, but with using the seaweeds, because they're sessile, which means that they're in the same place. They're attached to a substrate of some sort, so like a rock or maybe like the mud flats. And because they're in that same space at the same time, they take up the water within that same area. So it's a better analysis of the water in that specific spot over a longer course of time than, let's say, like DHEC, which goes in like once a day or however often they do it and takes that one sample. And that's only representative of that water quality during that specific moment. And because the ocean is super big, that water is fluctuating, the amount of salinity, its temperature, and its distribution. So it's not as good a representation as potentially seaweed could be.
That reminds me a little bit of a lot of times in freshwater systems, you'll look at macroinvertebrates, right? So the larvae of dragonflies or, various organisms that have a stage where they live in the water and then they emerge out to be whatever. And the types of invertebrates that you can find amongst the leaf litter in the bottom of a stream are really indicative of the water quality in that stream, because there's some species that can handle really high levels of pollution. There's some species where they will immediately die if they're exposed to those kinds of toxins. And so if you find something that's like super fragile and it's just living in abundance, you know that water is really good, clean, and safe. But if you can only find like black fly larvae, no bueno. It's not good. I really like looking at studies that use what is already in the environment as an indicator of water quality or of environmental quality, because I think it's such a more efficient way of determining how the ecosystem is doing as a whole. Yeah, that's a really, really cool project.
Now, Emily, you mentioned with the horseshoe crabs, you've only looked at, what'd you say, like 2 tanks of the horseshoe crabs at like 30 degrees Celsius?
That's the hot water one, right?
Yes.
Yeah.
Yes.
So obviously, I'm sure you want to look into this more. Do you have plans to study this more going forward? Is it just kind of you get to study it during the REU, but then you're done?
So I have 5 tanks total, and I've only gotten to processing 2 of those tanks, hence the 2 tanks. But I am interested in pursuing this going forward. I really love the research aspect. I love taking care of the crabs, which it's a lot of work sometimes. It's like I have 20 pets, but
They're kind of like dogs in a way.
What do you feed them?
Oh, goodness.
It's a good question.
This one's funny. So I feed them shrimp. Our facilities manager, Pete, he's had horseshoe crabs in the past. And he says, oh, you can either just drop them in the tank and wait for them and get out the extra so the water quality doesn't deteriorate, or you could grab them and shove the shrimp right in their mouth. I was like, Pete, you're crazy. I'm not going to hand feed my crabs. I hand feed my crabs. Pete was like, sometimes they spit them out, but I found that if you peel the shrimp in advance, they don't spit them out. So
They're high maintenance.
You have pampered crabs.
Yeah.
I
That's
know.
so considerate.
I know. So 3 times a week, I go into the lab and I peel my shrimp.
And you have very grateful horseshoe crabs for it. I want you to do a study on like the quality of blood on hand-fed horseshoe crabs versus like the ones that you don't hand feed. I think that would be funny, just to see if you can get commercial crab harvesters to be like, oh no, guys, this is artisanal hand-fed horseshoe crab blood.
Oh, yeah.
The nice thing about hand feeding is that I make sure everyone eats. Otherwise, it's kind of like, I don't know who's eating and who's not. So I want to make sure that I have nice, healthy crabs.
I totally get that, because when I worked at an aquarium previously as a scuba instructor there, we had horseshoe crabs in with kind of our main reef tank for a while. And we had a ton of porkfish, which they're not aggressive, but they're certainly not shy. And so anytime I would try to feed the horseshoe crabs, the porkfish would steal it. And so I would have to like kind of lift them up and put the food underneath them and then kind of hold them to the bottom until I was sure that they had grabbed it and put it in their mouths, and then I could leave them. So like I totally get it. Horseshoe crabs are like more finicky than you would expect an organism that's been around for that long to be.
How do you know who's who?
So the first thing I ever did with horseshoe crabs was go out with the South Carolina Department of Natural Resources and tag them. So they come up to the shore in large quantities, and the Department of Natural Resources will capture them, put a tag in them, and release them. It has little information on the tag. Okay, call in this number. If you see a horseshoe crab on the beach, please don't touch them. They're an endangered species, and human interaction isn't great for them. But if you see the tag number, go ahead and call it in, because the Department of Natural Resources loves that. But you essentially take a drill and you go through their carapace, and you just stick this little tack-like thing in there, and it's like swirly like a screw, and you stick it in there and it won't move. So yeah.
Cool. That's awesome. That's really, like, you're making me want to go play with horseshoe crabs, but it's not good for them, so I won't.
Yeah, we all tried to find them the one day, and it's a lot harder than you think.
Sophie and I were like into our waists,
Shuffling
in the water.
through the water.
Oh my
Trying not
gosh.
to step on stingrays.
And you like can't see the bottom, so you're like, I really hope that's horseshoe crab and not something worse.
And if you bump into one, you've got to go for it. If there's a second hesitation, they're fast.
Yeah.
They don't mess around, they run.
And I did capture all of the ones I'm using for my study. So
I love
Nice.
that. How long did that take?
Too long. A long time.
We were out there for like, I don't even know how long, an hour, just that one day looking, and came up with nothing. So
I would go out twice a day for at least 15 days and do that.
Wow.
So yeah, it's a lot. It's nice because in my past research projects, I've just worked on other people's fish or other people's samples. And it's like kind of cool that I've like hand harvest all of my crabs.
Your crabs.
But it was a lot of work.
the field work is like the best part. It gets tiring after, but it's fun throughout to be like really out there and
That's cool.
catching your own stuff or getting your own seaweed.
Yeah.
I don't really realize like how much work goes into field sampling. I don't think like a lot of people in the REU have helped other people with their sampling. I've helped multiple people, and it's interesting to see what other people do, because I'm like, oh, they're just getting mud samples, and then I'm out literally waist deep in mud. Like, am I ever gonna get out of here? I don't know.
Will I ever be clean again?
No, the answer is no. But it's cool to see how everybody collects their samples in such a different way. I like it.
Yeah. So talking a little bit about like the behind the scenes and all the work that goes into it. One of my favorite questions to ask researchers is what went wrong? We all have these great plans until you get punched in the face. So what happened with your projects this summer that you were not expecting that you had to kind of find a creative solution for?
Everything.
Yeah.
That sounds bad to say, but a lot. My sampling, like I said earlier, involves going out on the water. So first thinking that a summer in Charleston, there wasn't going to be a storm every single week. That was on me. Should have known better. Every week I'd be like, yeah, I'm going to go out and take my samples Monday. Monday rolls around, there's rain, can't go out in the water. Tuesday, rain again, and rest of the week, probably rain. And so now I'm about 2 weeks behind on my samples.
Oh, gosh.
But it is what it is. I can't change the weather.
That's science, baby.
Yeah.
It's a good attitude.
Breaking my glassware. I break a lot of glassware.
Oh, no.
It's kind of bad. But it's fine. You just order more and then hope it's not the super expensive ones.
Fingers crossed.
Yeah.
All right. What else? Who else had something go terribly wrong that you had to get a solution for?
It's okay if you can't think of anything. That's great. Good
It
for you.
wasn't. It's not necessarily terribly wrong, but there's definitely been a lot of changes in the experiment from what it was originally going to be. I had like different species that I was going to use, and I like started working on this project in the beginning of the semester, and I was originally going to do Porphyra or Pyropia. It's changed its name a couple of times, but that's what's used for nori. And I was like, oh my gosh, we can have our own nori from Charleston. But it's seasonal, so spring came and went, and that it's no longer in season. So that was out the door, and so now I had changed the species to stuff that's growing now, but also very weather dependent. And we did a pilot study a couple weeks prior to actually going out and collecting our samples. And some of the sites that we went to during the actual collection period no longer had the species that we were looking for, so we just had to keep picking new sites.
I know it's hard, but it's one of my favorite things about science that it's just like the show must go on. That didn't work out, so let's pivot. Let's figure out what else we're going to do with this, because we have this grant and we need to come up with something that we're doing with it. So I really appreciate the tenacity that comes along with the field practice of science.
Yeah. Just to wrap up, Emily or anyone else, do you guys have any recommendations for students who want to get into a REU program or to get into this field in general?
I would say I know a lot of people who go straight through their undergraduate year and they're like very one track, like I study biology because I want to be a biologist, or I study pre-med because I want to be a doctor, or blah, blah, blah. But I would recommend keeping your interests. I actually started on a pre-health track. I was interested in human health, and I was also a marine biology major, and everyone was like, how do those go together? And now I'm studying how marine organisms contribute to human health. And I think that made me a more interesting candidate all around. I think Sophie has something similar. A lot of people would probably think, how does public health go with marine biology? So if you're interested in something, don't abandon it because you don't think it will get you to where you're going as fast as you can. Efficiency isn't always as important as maintaining a lot of interests side by side.
I like that a lot because, like, my personal journey, I have an undergraduate degree in science and biology. I also have an undergraduate degree in theater. And here I am doing a podcast about science, right? So you can blend really weird things together in really cool ways. And that is a much more powerful combination than either one of those things individually.
Yeah, I would go along and say the same thing as Emily. I think passion is really important too, but just following that wherever that takes you. I started off with so many different ideas of what I wanted to do in my undergraduate degree. I think I came in with the intention of doing international business, and then for a second I was going to do sustainable fashion design, and then poli-sci, and then public health was where I ended up, and now I'm doing some marine biology stuff. So very much different tracks, but I kind of liked the idea of just following where whatever sort of sparked my interest and going with that, and having this untraditional background and having a lot of different interests has gotten me to where I actually wanted to go. So
That's awesome.
All I can say is if you want something, even though you don't think you're qualified for it, apply anyway. Like, imposter syndrome is super real, especially in science. Like, you're like, I don't know half of the things I'm supposed to know or what I'm talking about. And that's how I feel about 98% of the time. But somebody's going to believe in you. Dr. Podolsky decided to believe in me. Thank you, Bob. And here I am. So just apply for what you want. Believe in yourself, or fake it till you make it. That's what I do. And eventually somebody's going to take a chance on you and you'll prove them right.
I mean, we're all just faking it till we make it, right? So you're not faking it any worse than anybody else is.
Exactly.
You know, we're all just trying to become better versions of ourselves. Thank you so much for taking the time to be here with us. This has been an excellent episode. I'm so excited to get to share your stories. And until next time.
Thank you.
Thank you.
Thanks for listening to this episode of Conservation Connection.
If you enjoyed our podcast, go ahead and subscribe to make sure you catch every episode that we post.
We'd love to hear from you. So if you want to reach out, go to our website, lastchanceendeavors.com/contact and shoot us an email.
We love questions from our listeners, so if you heard something that you want to know more about, be sure to let us know.
If you've got a minute to spare, leaving a 5-star review on Apple Podcasts will help other conservation-minded people find the show. We'd really appreciate it.
A big thanks to the people working to protect our planet, and a big thanks to you for listening. Don't forget to tune in next week.